Photosynthesis is the formation of chemical energy stored in the glucose bonds and oxygen releasing in the presence of sunlight. Sunlight is a set of waves with different characteristics called spectrum. The radiation that reaches us has different wavelengths, and it's classified into three categories. Ultraviolet rays A, B, and C. They are not visible to the human eye and have various properties such as sterilizers and tanners. Visible range is the light that we can see and that has a variety of colors according to its wavelength. And the infrared range is the radiation that is responsible for the heat of the sun. Photosynthetic pigments are responsible for receiving energy during the photosynthesis and for the coloration of these organisms. These pigments are lipids bounded by some proteins located inside of the cellular membrane and have the ability to modify their bonds. Chloroplasts are in the leaves of the plants. These organelles can be as large as mammal erythrocytes and have reproduction resembling of bacteria. Chloroplasts are formed by two membranes separated by a narrow space. The external one has proteins called porins, and the internal one is waterproof and through the action of transporters. The photosynthetic reactions occurs in separate sacs in the inner membrane called thylakoids, and all of these structures are located in the stroma.
First stage of photosynthesis, light-dependent reactions. The sunlight is transformed into chemical energy from two carrier molecules, ATP and NADPH. Of the components of sunlight, there is electromagnetic radiation. All the reactions of the photosynthesis occur in the membranes of the thylakoids. They are organized into photosystems, one system collects light and the other carries electrons. Second stage of photosynthesis, independent light reactions. In this stage, the NADPH and ATP molecules synthesized are dissolved in the stroma that surround the thylakoid, and from there, they provide the energy required for the synthesis of glucose from CO2 and water. Calvin-Benson cycle or C3 cycle captures CO2 using RUVP, that is a 5-carbon sugar. Carbon fixation gives rise to 12 molecules or 3 carbon atoms of PGA. Synthesis of G3P transforms the PGA into G3P. RUVP regeneration is used to make glucose. 4-carbon root or C4 binds CO2 in 4 carbon compounds instead of 3. Second,
factors that affect photosynthesis. The external factors are light, carbon dioxide, temperature, oxygen, nutrients, water, and humidity. And the internal factors are RUBs, CO, activity, chlorophyll, and leaf color coloration. Artificial photosynthesis would allow us to store large amounts of solar energy, which could be used in areas where this phenomenon is not possible. Also, solar energy can be transformed into sugars to produce biofuels later.
According to the Encyclopedia Britannica, cellular respiration is the process by which organisms combine oxygen with foodstuff molecules, diverting the chemical energy in these substances into life-sustaining activities and discarding as waste products carbon dioxide and water.
Metabolism is a series of reactions, whether or synthesis or degradation, of molecules within a cell. This process is a network of reactions connected to each other and very regulated. The metabolism is divided into processes: exorganic reaction, which is catabolisms, endogenic reactions, which is anabolisms. Catabolism From the potential for degradation of organic nutrients, they realize energy for cellular work, and anabolism is they produce the new component absorbing energy applied to the functioning of the cell. In stage 1 is glycolysis, glucose, then pyruvate, and it gives to ATP substrate level. In stage 2 is acetyl-CoA, then citric acid cycle, and then chemiolysis. In citric acid cycle, it gives to ATP substrate level, and in chemiolysis, it gives 34 ATP and it's oxidative.
We're gonna talk about now the process of cellular respiration. As you can see in the diagram shown in the presentation, there are 3 steps for this process. First one, the macromolecules such as starch, triglycerols, and proteins are minimized or making smaller molecules. So we can go to step 2, which is that the molecules that were transformed into smaller molecules are now transformed into acetyl CoA, which mean which means acetyl coenzyme A. Now in step 3, the acetyl along with carbon dioxide will go into the Krebs cycle, so it can become a reducing equivalent, and its electrons will go into a transport chain. In this last step, it creates water, which is one of the waste products. Now I'm gonna show you the chemical equation for the process of cellular respiration. And as you can see, it's like I explained in the other slide. It's just the macromolecules transforming into smaller ones and being in a chemical equation themselves.
Amount of ATP produced. The total amount of ATP that a cell is able to harvest from each glucose molecule that enters glycolysis depends on the presence or absence of oxygen. ATP from one molecule of glucose is 30 ATP. ATP from one molecule of pyruvate dehydrogenase is approximately 15 ATP. ATP from one molecule of acetyl-CoA is 40.2 ATP, and ATP from one molecule of oxygen is 38 ATP.
Fermentation is a chemical process by which molecules such as glucose are broken down anaerobically. Louis Pasteur in the 19th century used the term fermentation in a narrow sense to describe the changes brought about the yeast and other microorganisms growing in the absence of air. He also recognized that ethyl alcohol and carbon dioxide are not the only products of fermentation. Fermentation is a metabolic process by which glucose or other 6-carbon sugars are converted into cellular energy, and the metabolite lactate, which is lactic acid in solution. In humans, lactate is transformed back into glucose in the liver. The term lactic fermentation is no longer used. In contrast, in microorganisms such as Lactobacillus, which uses lactose as a metabolic process. Fermentation. The route which glucose is broken is called glycolysis. If there is oxygen, then the process will be done by the cellular respiration. By this, the final product is pyruvate, but if there is no oxygen, the final outcome will be lactate.